Please feel free to chat to Lang about what I think is some very interesting and important work during the tea. And our final speaker before the tea is Kerryn Warren, and she's going to be telling us about using art to understand human evolution.
Hi, I just want to start by saying that nothing on these screens code for anything, so it was just a stylistic choice, if you will. This is also giving away the plot. How do I? Thanks. It's stable. So essentially I'm in this very strange discipline in the sense that we've heard a lot of people speaking about their data. I'm not going to speak about my data per se. I'm going to speak about my discipline more broadly. And they're saying things like we're showing all these models and we're doing all these statistics with their n equals hundreds and thousands. And when you're in a discipline like mine, you're actually pretty jealous of that because it's not unusual for us to have a sample size of 2 representing an entire species. We have very few fossils, is what I'm trying to say. And while we don't only have those on the board right now, Ian Tattersall once said that we could probably, if we didn't mind jumbling them up a bit, we could probably just shove all the human ancestors and cousins into the back of the van as to what we have. Not recommended in my discipline either. So this presents us with quite a challenge. We have to essentially either find more fossils or we have to really study the ones we have. And we try and do both, really, in order to inform on how to study, how to find more fossils, and then of course to add to our sample sizes and to understand kind of where we went wrong. So in the find more fossils scenario, we tend to rely heavily on mapping. And now with a lot of GIS software out there, that's great. But the big problem is that a lot of these fossils are found in developing countries. We're trying to collaborate between different institutions with varying resources. And so trying to put this all together is actually quite difficult. So we try as best as possible to use open source or freeware. And that's where sort of R comes in. And I've listed some things up here as an example. As I said, these don't code for anything, but our QGIS is quite useful because we use QGIS wherever possible because it's also freeware. And so a lot of people have that expertise, but in order to do some kind of real world data analysis on it, it's useful to have a statistical package as well. And this was done, unfortunately it wasn't done in R, but it's a good example of what could be done in R, in the sense that people had 3D scanned the cave of Dinaledi. I don't know who's heard of Dinaledi. Because a Homo naledi was essentially found by Lee Berger a couple of years ago. And it's just a whole bunch of hominins at the bottom of this giant dark cave system. And it's not a very easy cave system to navigate around, so how did they get there has been quite a big problem, if you will. And so trying to figure out where they are, how they got there, has actually been quite questionable. So now they've sort of figured out the orientation and to maybe find new caves that are similar in the system. What has been done in R has been us taking our digging areas, where we're digging, because we do dig, where are those little people with the brushes and sometimes the spades, trying to get to fossils, essentially in the ground. And it's actually very useful for us to find out where they are in terms of layers and to understand the structure of those quite nicely. And so by using GIS software and mapping and overlaying that with 3D models, we can have a really good idea as to the geology and where the fossils are found. There have also been some more specific kinds of packages that have been designed, such as ZooArc, which essentially just says to us how well the animals or the fossils of animals, not necessarily humans, in the archaeological record survive. And of course, we can use it for dating. And now this gets a little bit more to my stuff. Me and people in my lab, we tend to use 3D scans of skulls, not always humans or hominins, but sometimes animals as models for the humans and hominins. And the reason we do that is fairly obvious in some ways. In the sense, if we look at this room, we all kind of look different, right? And if I had to just take 2 of you and said, this is humankind or modern humans, it might be a little biased. In fact, even if I had this entire room, it might be quite biased as to what modern humans look like. So we try to use animal models wherever possible by sort of fitting those 2 individuals into a variation of what we get in animals in general. And this means that we can actually toggle or play around with shape space more generally and try and get an idea as to sort of how big certain individuals might be able to get within the species that is only represented by those 2 individuals. And we can do some pretty cool stuff. This was done by a friend of mine, Lauren Schroeder. She looked at what we call genetic drift, which is random chance just affecting the shape of the skull. And she found that actually there are certain things that affect the head that seems to just be random chance, which means it's not selection, survival of the fittest, that is driving the change in the shape of the head. It's actually just chance. And last slide, I'm looking at animal models in the sense of mice. I'm looking at different mouse species and hybridizing them and trying to see if there's a consistency in the pattern of the shapes of their heads using some R packages such as Geomorph. And then of course you can see I'm rocking the ggplot right here. To try and see if there's any consistency in shape change that we can see in hybrids. And as a random application, we might be able to use this to detect human Neanderthal hybrids in the fossil record. So watch this space. So thank you to the organizers.